and as Madeleine said, I'm not a vet, and I'm going to talk to you about how to make use of monoclonal antibodies in vaccine design. Before starting that, I just need to mention that the company I'm working for has been last year acquired by a bigger organization, Vir Biotechnology, which has the headquarters in San Francisco, and we kept our site on antibody discovery and development in Switzerland, in Bellinzona, and the Vir Biotechnology is funded by H2020 mainly, but also by the Bill and Melinda Gates Foundation, and that's why there is an ongoing work on the development of CMV-based HIV and TB vaccines, and in general the company is aimed to develop new ways to treat infectious disease with antibodies and other means. And the current focus is related to the development of treatment for respiratory infection, in particular influenza and RSV, as well as for chronic infection with a focus on HIV, TB, and chronic HPV. I would like to start back with some historical perspective on the use of antibodies for serotherapy. This goes back to the use of horse anti-sera to protect from diphtheria and tetanus toxoid, and this led also to the first Nobel Prize in medicine to Von Behring in 1901. And later, about 20 years later, there was the first attempt to use convalescent plasma to treat severe influenza infection during the Spanish flu, and this has been then used afterwards in many other occasions for avian flu as well as for SARS, with some, in some cases, some clear kind of efficacy, but it was never formally proven. And on top of that, since the 60s, there was the development of specific purified human immunoglobulin products, like, for instance, rabies immunoglobulins or CMV immunoglobulins, which have been purified out of human blood or blood from horses, for instance, to treat specific diseases. But clearly, another important milestone in the use of antibodies in medicine has been the ability to isolate monoclonal antibodies, and this was made possible since 1975 with the development of the hybridoma technology, which was developed by Köhler and Milstein. They also won the Nobel Prize in 1984, and interestingly, they didn't patent this technology, so everybody has been using this technology freely until now. And this led to the development of the first monoclonal antibody used in humans, which was an anti-CD3. This is not used anymore. This entered the clinic in 1985. And in the case of infectious disease, the most important and still the only one which is largely used antibody against an infectious disease agent is Synagis, which was introduced, which was approved 20 years ago, in 1998, and it is an antibody against RSV, and I'm going to talk a little bit about that. And nowadays, there are more than 40 antibodies that are licensed to treat inflammation, cancer, and infection. But as you can see from this updated analysis on the targets of monoclonal antibodies, the large majority of the targets are in oncology and inflammation, with even more antibodies on the market against the same target, like for instance for CD20, there are nowadays 4 antibodies on the market. In infectious disease, which are those highlighted in red, there are nowadays only a total of 5 antibodies approved, of which palivizumab, as I said, from 1998, RAB1, which is an antibody against the rabies virus, which is just approved for use in India, which was approved 2 years ago, bezlotuzumab from Merck for the treatment of Clostridium difficile recurrent infections, and 2 monoclonal antibodies against the protective antigen from Bacillus anthracis, which have been approved 2-3 years ago and used for stockpiling in case of bioterroristic attack. Clearly, one of the difficulties in the development of effective anti-infective monoclonal antibodies was also related to the difficulty of isolating highly effective antibodies out of immunized mice or also from phage libraries. And I will come back to that in a second. I just wanted to provide a brief overview of which are all the possible mechanisms of action of anti-infective antibodies. Obviously, on top of that, you have the neutralization, which can be true for the blocking of viral entry or viral fusion, the neutralization of the toxin activity with multiple mechanisms of action. But the Fc piece of the antibody can also trigger the engagement of the immune system through killing of infected cells, for instance, by NK cells, the activation of the complement, which can cause the direct killing of infected cells or the direct killing of the pathogen, phagocytic activity of phagocytosis of infected cells, or again, the phagocytosis of opsonized bacteria, for instance. And other mechanisms also related to the fact that antibodies can provide an aggregation of pathogens, for instance, viruses, reducing the overall infectivity and growth, and the formation of immune complexes can also promote the clearance of the pathogens and also trigger a better immune response. So it's a large set of overlapping mechanisms that can be also somehow modulated, and this is possible through Fc engineering. And nowadays, for instance, in the case of the development of monoclonals for prophylactic use in humans, there is the possibility to introduce the point mutation in this region of the Fc, in the CH3 region of the Fc, so to be able to prolong the half-life of antibodies up to 80, 90 days. And there are already antibodies in the clinic that have shown this level of half-life. And obviously this would allow, in principle, to provide passive protection for quite some time with a single dosing of the antibodies. At the same time, as I said, all the Fc effector function can be modulated in one way or in another way, in the sense that it can be the binding to, for instance, all the Fc gamma receptor as well as complement can be abolished by the introduction of just a few mutations up there in the CH2 region. And at the same time, glycoengineering, these are the glycans in the Fc piece, and other mutations can also be used to modulate the interaction with the whole set of activatory and inhibitory Fc gamma receptors. And there is really a large set of mutations available out there that can be used according to the specific needs. One big problem is that sometimes we don't know well how to modulate them. So, and obviously, it's very important to have very reliable preclinical animal models to test this. In our lab, we developed over the years two approaches to isolate human monoclonal antibodies out of the two possible sources of antibodies, which are memory B cells on one side and plasma cells on the other side. The first method, both methods relies on cell culture of cells and the ability to secrete the native antibodies in the culture supernatant and test the antibodies for their activity, and in what we call mainly, in most cases, an agnostic approach. The first method is based on the efficient immortalization of B cells with Epstein-Barr virus coupled with the toll-like receptor agonist that increase highly the efficiency of immortalization. In this case, the clones grow up and produce a lot of antibody in the soup. In the second method, we found out a way to keep plasma cells alive long enough in culture to produce enough antibody to be tested. And in many cases, we can screen the antibody repertoires provided by the generation of libraries with the two methods with many parallel binding and functional assays like binding, neutralization, and so forth. In most cases, we don't necessarily need to have purified antigen to start with. And with these two methods, over the years, we have isolated antibodies against a very large set of viral and bacterial targets. Some of them are already in the clinic, in particular antibodies against human cytomegalovirus, a neutralizing antibody against influenza A, and more recently, it will start soon, the phase 1 an antibody against Ebola virus. Is it me? No. But today, on top of, as I said, making use of antibodies as drugs, I would like to talk to you about the possibility of making use of antibodies for other purposes. We have used them successfully, and we have had an interesting collaboration last year with Eva Harris in Berkeley on the development of serological diagnostic assays to distinguish between Zika virus and dengue virus infections, as well as making use of antibodies to identify cellular receptors for complex pathogens. We successfully found the receptor for cytomegalovirus on fibroblasts, which was found to be PDGFR alpha, and more recently, this is about to be published, we also found the receptor for the pentamer. But I would like to talk to you about how to make use of antibodies for vaccine design. As we know, there are major human diseases for which yet there is a great need for the development of vaccines. I mentioned cytomegalovirus. For sure, dengue is still, despite the vaccine is on the market now from Sanofi, I think there is clearly need to do something better than what it has been developed. Influenza, for the need to develop a universal flu vaccine, and RSV, which is what I'm going to talk to you about. This is an interesting overview by Rino Rappuoli. He was working at Novartis, now he's at GSK, on how vaccines have been developed until now. So there was the classical vaccinology approach, which was based on the use of killed or live attenuated vaccines, and later also the use of subunit vaccines, but this was a largely kind of empirical approach. And many years after, Rino Rappuoli pioneered the so-called reverse vaccinology approach, in which essentially antigens from complex pathogen, in this case bacterial targets, were selected in silico, expressed and selected in animals for the best, to come up with the best immunogens. And this kind of approach led to the development of the MenB vaccine, which was approved by EMA in 2012. But Rino, in this review, presented what he called reverse vaccinology 2.0, which is based on a much more complex set of approaches that comprise genomics, high throughput protein expression, the use of monoclonals, which is what I'm going to talk about, B-cell repertoire deep sequencing, structure-based antigen design, and in a way this was started in 2013 with RSV. With the development of the first kind of a new RSV vaccine candidate. But before entering and talking about antibody-driven vaccine design, I would like to talk to you to tell you briefly about the fact that we need to develop the right type of antibodies, and we have had cases in history in the past, but even more recently, in which it is clear that we have to try to develop the right type of antibodies. In 1968, the first attempt to develop an RSV vaccine failed. And actually, in this case, the formalin inactivated RSV vaccine led to an enhanced disease in children. And this has clearly altered the development of an RSV vaccine for almost 40 years. And this enhanced disease, which was being studied a lot, was likely related to the development of weakly, poorly neutralizing antibodies that form immune complex deposition and complement activation in inflamed airways. And at the same time, this was also linked to an exaggerated inflammation and Th2 biased immune response, as it was well studied and described by Fernando Pollack. I think another example, as I mentioned earlier, is represented by dengue, in which we know that naturally immune donors are typically protected when from a homologous infection, from reinfection with an homologous virus from the same serotype. There are 4 serotypes for dengue, but there is a clear and well-demonstrated risk of developing severe disease, in particular dengue hemorrhagic fever, when individuals are infected with a different serotype. And this has been shown and more recently demonstrated to be linked to the antibody-dependent enhancement mechanism. And this is an example of what it means, ADE. You can have and isolate a lot of monoclonals against different viral antigens. In this case, the domain 3 of the E protein, the domain 1 and 2 of the E protein, or the pre-M antigen. And you can test the antibodies in vitro against dengue infection on competent cells, like in this case, vero cells. And you can see that when you increase the dose of the antibody, you have a clear neutralization, so no infection. But when you perform this on cells displaying Fc gamma receptors and cells that are not naturally competent for infection, you can clearly see that you have a bell-shaped type of infection in which in a certain range of concentration of these antibodies you have enhanced infection in vitro. There is a vaccine now on the market that has been developed in the last 15 years by Sanofi, and this is called Dengvaxia, and it is a tetravalent live attenuated vaccine, which is based on a yellow fever virus backbone. In the phase 3 trial, they found that there was a 5 times higher incidence of hospitalization due to dengue in vaccinated young individuals. And indeed, the licensure of Dengvaxia was limited was limited to individuals above 9 years of age, and was also recommended to be used in population where there is a high seroprevalence of dengue, which is a little bit contradictory for a vaccine, because it has to be used where there is already a lot of existing immunity. And recently, because of a quite sharp increase in the number of hospitalization in Far East, in Thailand and other countries, they had to change the labeling of the vaccine to actually limit the use, not just based on the age, but also on the fact that the vaccine has to be used in people who are already immune. So in other words, it is probably a good vaccine for a boosting, but not a very good vaccine for the priming. Despite a lot of concerns that were raised from the data from this phase 3 trial, they went up with the development of this, and it has been quite recently demonstrated and formally demonstrated indeed that the severe dengue disease in humans seems to be related to the antibody-dependent enhancement. This has been nicely demonstrated in a large cohort study in Nicaragua in children by Evaris, and this was published last year in Science, where they have shown that there is the risk of severe dengue disease is very high within a narrow range of pre-existing antibody titers. This is highlighted in red here. This has been a formal demonstration of the ADE in humans. And so there is clearly a need to develop new type of dengue immunogens where the risk of having these problems is diminished. And this is probably possible through antigen engineering and trying to maximize the development of potently neutralizing antibodies. I'm going to talk now about 3 different approaches in which we can make use of antibodies for the development of new vaccines in what we have called analytic vaccinology, which is highlighted here in the identification of relevant antigen on complex pathogens. The second case is about structure-based vaccinology, so making use of antibodies to identify the right structures to be developed as immunogens, and finally to make use of antibodies against specific domains or epitopes of proteins to focus the immune response against those regions. So the first the first case I would like to talk to you is about the development of CMV vaccines, new CMV vaccines. This project started trying to answer which are the most relevant target antigens that are targeted by potent neutralizing antibodies. Cytomegalovirus has a large set of glycoproteins that are involved in the entry process, like the GB, fusion protein, the dimeric GHGL, the pentameric structure. And our project here started from in an agnostic fashion, in trying to isolate antibodies for their ability to neutralize, in this case, the infection of CMV on 2 target cell types, so in particular fibroblast and epithelial cells here. So we found and isolated antibodies based on the parallel neutralization of infection against these 2 cell types, and just subsequently we tried to find out what was the target antigen. And we found that there was a class of antibodies down there, and initially we didn't know what the target was, that were very, very potently neutralizing the infection in the sub-nanogram per ml range. They were really super potent antibodies by different means and different approaches based on the staining of cells, cells transfected with the CMV genes alone or in combination, and cross-competition studies. We found out that these antibodies in red over there are targeting this pentameric complex on the surface of the virus, and we have identified that there are multiple, now more than 10 distinct antigenic sites have been defined on this huge structure over there. And we try to develop this protein by getting rid of the transmembrane piece as a recombinant protein, and others after us have been doing the same, and trying to develop this as a new vaccine, and tested this initially in mice at the different doses with different type of adjuvants. In this case, we have used a Rsv adjuvant, and shown actually that this kind of vaccine can elicit sky-high titers of neutralizing antibodies in mice, 10,000 higher levels than what you can measure in convalescent patients upon natural infection. So in other words, in this case, you can do much better than natural infection. And there is now a clear interest in developing a pentamer-based vaccine, in particular for the possibility of inducing a protective immunity, reducing the risk of congenital CMV infection, which in humans is a big problem, and it is the most important viral congenital infection. In this study, we have also investigated, similarly to what we are doing in humans, the monoclonal antibodies that are induced by this immunization by using the classical hybridoma technology. And we found out in parallel that the GB protein, which is a fusion protein that was used in this study, elicited a huge number of antibodies, but just a minority of them were able to neutralize the infection. Whereas that was exactly the opposite for the pentameric antigen. And we think that the issue why the GB vaccine induced just a minority of neutralizing antibodies has to do with the structure of the GB, which when we use it in this study, it is a structure which is in the so-called post-fusion state. We have also mapped the antibodies and found out that the antibodies induced in mice well recapitulated what we have found to be developed in humans upon natural infections. So, as I said, other companies, and there are at least 2-3 big pharma who are trying now to develop this kind of vaccine in the clinic. But clearly the prompt and the start for all of this has been made through the use and the understanding of which were the most potent neutralizing antibodies. So the second case, which is highlighted here, is based on, it will be related to the development of an Rsv vaccine. Rsv is an important disease for infants. It is the most important cause of death related to lower respiratory viral infections worldwide. And it is not just a problem in children, but also a problem in the elderly, as well as in immunocompromised patients. And as I said in the beginning, there is already a monoclonal antibody on the market, Synagis. The name of the antibody is palivizumab, which is given to premature infants at 50 milligram per kilogram intramuscularly. And nowadays it is recommended just for premature infants with less than 29 weeks of gestation, born from. And however, the use of this antibody is rather limited by the very high cost of the treatment, which is not really related to the cost of goods, but to the very high pricing of the product. And the dosing regimen is also not very convenient because it has to be given monthly for 5 doses. And so in all, it is clear that palivizumab use is not cost-effective for use in a lot of non-high-risk groups, which, however, represent the majority of the cases. And with palivizumab, it is clear that the protective level of the antibody has to stay above this red dotted line, so which is about 40 micrograms per ml to protect. And actually, protection in this case means protection from severe disease, does not mean protection from infection. So it's something different from providing sterilizing immunity. So palivizumab, as well as other antibodies, were originally developed using an antigen, the fusion protein of RSV, which has been found to be in a conformational state, which is the so-called post-fusion state. So this is not really the right target for neutralization, because this is the structure that is formed when the fusion event is occurring or has already occurred. And And the post-fusion structure, which is this very elongated protein, has been also used and developed initially as a potential vaccine by Novartis and also by Novavax. And Novavax has phase 3 vaccines in development in humans based on this structure and based on the assumption that since palivizumab is working and it is targeting a site over here, the so-called site 2, then this is supposed to be a good antigen. But I think that all in all has been proven to be a wrong assumption, because clearly we should be able to target mainly the pre-fusion structure of the antigen to elicit more potent antibodies. And I think that the best work that pioneered this concept was back in 2012 by the group of Melero and colleagues, where they found that, and for the first time they were able to develop a first form of a stabilized prefusion F protein. And they demonstrated by using the post-fusion F protein that this was not able to remove the neutralizing activity of F-specific antibodies from the serum of immune people. And they found out that most of the potent serum neutralizing activity was instead accounted by prefusion-specific antibodies. And just a year after, my group been working on the isolation of a lot of monoclonal antibodies, and the isolation, like I showed you before for CMV, was based on isolating antibodies based on their neutralizing activity. And when we tested them against the F protein in the pre-fusion form or the post-fusion form, we found out that the large majority of the neutralizing antibodies were specific for the pre-fusion, which is obviously relevant. And at the same time, the group of Peter Kwong at the NIH, by using one of these monoclonals directed against prefusion-specific monoclonals, he was able to stabilize and get the crystal structure for the first time ever of the prefusion F protein of RSV. And he defined this site as a site 0. And in another paper on the same issue, he also published on how to further optimize and stabilize this prefusion F protein by introducing these stabilization motifs like a fold-on trimerization domain. They introduced a lot of cysteine pairs to form and stabilize the trimer, so to form interomonomeric disulfide bonds. They introduced some so-called cavity-filling hydrophobic substitutions. This led to a stabilization of the molecule. And in all of these tests and all of this engineering of the antigen, I don't go through the details here, they have always made use of monoclonals to probe the stability and the potential immunogenicity of the antigen, which was further engineered also to be stable under very harsh conditions, so low pH, high pH, high and low osmolarity, and freeze-thaw cycles, so to end up with a very stable type of antigen. And they demonstrated in mice and in macaques that this kind of stabilized prefusion antigen could induce a level of neutralizing antibodies much higher as compared to what were found to be elicited by the post-fusion F protein. I would like to tell you about, as a follow-up of this story, on our collaboration that we started with Peter Kwong and Geraldine Taylor just after this publication. So we were considering as a target also bovine RSV. We know that there is a need for the development of an effective commercial bovine RSV vaccine. I think I don't have to tell you about the importance of bovine RSV and the fact that still the existing vaccines are suboptimal. So in this case, we have done a little bit the opposite of what you usually do. We started from human RSV and we went back to an animal virus, and we tried to perform the same kind of engineering on the F protein of bovine RSV. In this case, you see the differences that are out there between human RSV and bovine RSV, and the differences are highlighted here in orange. And through, again, a combination of stabilization motifs, in this case also including an additional change, which is the fact that we removed the linker region, so we ended up with a single chain protein, so Typically the F protein is cleaved in F1 and F2. In this case, this is a single chain, and at the same type of changes that were I was talking about before. And again, using MABs, we were able to end up with a series of constructs that are shown here. In total, 8 different constructs, and these are actually the shape of these constructs by cryo-EM that were tested for their ability to be bound by a set of human monoclonal antibodies cross-reactive to bovine RSV and targeting distinct antigens, as well as this other kind of physical stability test. And we initially tested all these 8 antigen in combination with post-fusion F bovine RSV F protein in mice. And we immunized mice with 10 microgram of adjuvanted poly-IC protein, 50 microgram per animals, and found out indeed that the pre-fusion constructs were able to induce much higher neutralizing titers as compared to the post-fusion F immunogen. And of this, we decided to select and elected the DS2V1, this one in red, as the potential candidate for further development. And that's why we decided with Geraldine to test this antigen in calves. And in this case, calves received 50 microgram of this antigen. And in parallel, we tested also post-fusion F protein. And the animals were primed and boosted 4 weeks after. And we have checked the level of neutralizing antibodies developed over time. And as I will show you, these very same animals have been also challenged afterwards with bovine RSV. And we found out that after 6 weeks, the level of neutralizing antibodies reached levels close to 100,000 titers. And as compared...
What is the post-fusion?
The post-fusion is the blue. The blue is this one, which is about more than 2 logs lower in terms of neutralization. And typically in RSV, in humans, there is this concept of considering the 1 in 100 like the protective titer, but it's just an assumption based on the palivizumab data. But it is clear, apart from that we are clearly, we have reached levels far higher than what it has been ever seen with other kind of immunogens. And when the animals were challenged with bovine sorry, this is about, before coming to that, we have also tried to characterize the serum of immunized animals for the presence by performing competition ELISA assays of antibodies against targeting different sites on the pre-fusion F protein. So we have used as probes a set of different monoclonal antibodies that for which we know the epitope, and shown that the DS2 vaccine elicited a high level of antibodies, neutralizing antibodies directed against multiple sites. So the response is not just focused against a single site, but against a large set of antigenic sites that have been demonstrated to be targeted by highly potent neutralizing antibodies. When the animals were challenged, it was really nice to see that there was no detectable at all RSV in nasopharyngeal secretions in animals that were challenged on week 8, whereas the level of virus between the control group and the animals receiving the post-fusion was similar. And that was also paralleled by no detection of virus in the lower respiratory tract in any part of the lung that was tested. And on top of that, this was also paralleled by almost a complete lack of lung lesions or cellular infiltration. At the same time, in terms of disease induced by the infection by bovine RSV, we have also found in terms of clinical score, which is largely driven by the respiratory rate and temperature, there was a clear trend for the DS2 to protect animals from disease. And all in all, I think this data highlighted the potential to develop this as a new bovine RSV vaccine. It's, I have to say, not easy to find traction to develop this type of vaccine And we have been trying, and if any one of you is interested to talk more about this, because this is clearly not the focus of the company I'm working with, but I think there is a potential to develop a new type of vaccine following up what it is now done in humans, because there are at least 2 vaccines in development for human RSV based on this kind of concept. So it can be done also for animal viruses. The last example I want to talk to you is about influenza. So you know that there are multiple subtypes of influenza. We have had in the last century about 5 pandemic events by 3 different subtypes. The last one is the swine flu from 2009. Other outbreaks from different subtypes have been also documented. I think I can skip this. And we know that the majority of the neutralizing antibodies that have been described and that represent also the way in which we define correlates of protection for flu are those directed against the head of the hemagglutinin over there. And they are the antibodies that are so-called HI positive, so those that can block the hemagglutination. Whereas in the last now more than 20 years this year, it has been also found that neutralizing antibodies can also be found to be directed against the more conserved region of the stem of the hemagglutinin. The very first study was back in 1998. It was clearly a study that was neglected for about 10 years when the first the group at Crucell found the first antibodies against the stem from humans, and other groups after that from phages have found the same. Our group in 2010 it found out it from out of vaccines and so forth and so on. And this first group of antibodies were found only to be able to cross-neutralize influenza A subtypes belonging to the group 1, but there is also a group 2 which is represented in humans by H3N2, and other antibodies targeting the group 2s, all the group 2 viruses by eating the stem region were isolated afterwards. We were the first to be able to isolate an antibody able to cross-neutralize all group 1 and group 2 subtypes, and this was possible by isolating these antibodies after the 2009 pandemic virus. So we found out a very high frequency of cross-reactive antibodies in plasma cells, and we found out very rare events and very rare antibodies able to cross-bind, in this case, H7, which is a group 2, and H5, and out of this we developed the first antibody, which is called FI6. And just a few years after, we found out another more potent antibody that is now in the clinic, which was further engineered and optimized in vitro for potency against both group 1, H1N1 in this case, and then H3N2. And this antibody was also shown to target a large set of animal viruses. And importantly, this class of antibodies targeting the stem inhibit the entry by blocking the fusion, but I think there are other mechanisms of action that have been shown to be important. One is the fact that they can block the maturation of the virus because they prevent the cleavage and the maturation of the HA extracellularly by preventing the proteases from cleaving the HA. And they have been also shown to be highly active by making use of effector functions. And this is an example. So we have tested this FI6 antibody prophylactically by engineering it to remove all effector function with these LALA mutations. And we found that the antibody activity was significantly reduced by this modification. And when we did the test in vitro, we found out that the antibodies targeting the stem region are very effective in promoting the killing of infected cells by NK cells, whereas antibodies targeting the head over there are almost not at all active in this respect. And this is related to the mechanism of action. And this has been confirmed in other studies by others, showing that clearly the activity of anti-stem antibodies relies a lot on the contribution by activating Fc gamma receptors. And more recently, it has been shown that this is not just because of NK cells, but alveolar macrophages play a very important role in a protection by anti-stalk antibodies. And this obviously has not just to do with ADCC, but also with the phagocytic activity that is exerted by macrophages. There is also the concern in flu, and this was due to the study in pigs, that there could be a risk for enhanced disease by antibodies targeting the stem. This is a study back in 2013. And in this study, they demonstrated that in pigs vaccinated with the whole inactivated H1 and 2 pig strain, and then infected intratracheally with the pandemic H1N1 virus, they found out a very severe enhanced disease in animals. And they found out this to be correlated to antibodies against the stem region, but antibodies that were able, even in vitro, not to neutralize, but actually to enhance the fusion of the virus. And we tried to understand whether this was also a risk for anti-stem neutralizing antibodies, and this is another collaboration we have had with the Pirbright, and in particular with Elma Chilian. And we tested the FI6 antibody in pigs, trying to see whether we could see any sign of disease enhancement, and we didn't. It's also true that we found out that the pig FC receptors do not bind to human IgG1 at all. And obviously that means that the effector functions that might be important for the activity are not measurable in these animals. But we also found out that when the antibody was administered 1 day after infection, there was a trend for a better outcome in terms of gross pathology, but not really a strong protection. So we believe that the lack of effector function over there probably played a role in not seeing high efficacy, despite the fact that we could measure pretty high levels of neutralizing antibodies in the serum. And this, I think, suggests once more that in vitro neutralization by antibodies is just one side of the coin, and there is much more than that. And I think we still don't fully understand which are the best correlates of protection for flu. And I'm coming to the end. So, and this is about the attempts to try to develop new immunogens for flu in order to try to focus the immune response against the stem and avoiding the response to the highly variable head region. And this was pioneered in the beginning by Peter Palese by making this headless hemagglutinin, but they didn't really work out too well. They were not well stabilized, and probably the real structure was screwed up when they were used as immunogens. And he followed up on that by making chimeric hemagglutinins in which the head was actually replaced by the head of different subtypes, avian subtypes, so to try to exploit in a positive fashion the original antigenic scene, so to make and try to develop a natural mechanism of immunofocusing. And this is based on the concept that we already, most of the individuals are already immune and already have developed anti-stem specific B cells, and they can be boosted by sequential heterologous vaccination with this kind of chimeric construct. And this is a kind of approach in development. And more recently, independently, two different groups at the NIH over there and at Janssen and Krusell developed two forms of stabilized headless hemagglutinins, and they have shown that this can provide protection against heterologous strains in mice and also in ferrets, despite really not providing a really high level of neutralizing antibodies. So most of the antibodies elicited by this type of vaccines, which were also developed through engineering, as I said before, and using the neutralizing monoclonal anti-stem antibodies as probes. Despite that, the majority of the antibodies induced were not really neutralizing, meaning that probably there is more work needed to further stabilize these structures. Nevertheless, this specific kind of immunogen here, which is called 4900, is now in the clinic. They have done a phase 2 in humans by Janssen with the support of BARDA, and they are trying to develop this as a kind of new wave of universal influenza vaccines. And I think that on top of that, there is more work to be done, and I think that there are other issues to be considered, and that in particular, for instance, for the development of an HIV vaccine have to be combined with the development of new immunogens. I think it's going to be very important to understand which are how complex are the developmental pathway of antibodies to induce mature and high affinity antibodies. This seems not to be that an issue for RSV, but it is clearly a big issue for HIV, where it has been shown that the very good broadly neutralizing antibodies have to accumulate a really high load of somatic mutations. And that obviously complicates a little bit the vaccine design, because you have to try also to develop a way to induce that very same level of maturation with immunization. At the same time, obviously, and this is also a very much evolving field, there are novel platforms to deliver the immunogens through VLPs, nanoparticles, and nowadays there is a lot of hype about this, the possibility to deliver this antigen through DNA or RNA type of vaccines. Obviously, I'm not really an expert, but adjuvants are obviously are a key, and it's also important to assess whether the antigens that are stabilized in vitro are also stable in the presence of adjuvants that sometimes can be can have some destabilizing characteristics on the immunogen. And I think that it's also important, at least for some diseases, to be able to have some more rapid and small and hypothesis-driven development in the clinic. And I think that with that I'm done, and I would like to acknowledge all the people in my lab, the very fruitful collaboration at the Institute in Bellinzona, in particular Antonio Lanzavecchia, was my mentor, and I think the great collaboration I have had and I'm continuing to have with the Pirbright Institute, a collaboration with the NIH and also MedImmune on the flu part, and all of you for the attention.
Davide, I will start. Now, I have a question about how do you avoid this hypersensitivity reactions? Okay? With this old, let us say, formaldehyde inactivated vaccines, RSV, and the other ones you mentioned. Now, if I understand well, it's immune complex mediated Arctus reaction, type 3 hypersensitivity, if we agree on that. Now, is it the idiotype? Is it the affinity? Is it the amount, antigen, antibody, which all influences whether or not you will have this hypersensitivity reaction? If we know how it works, how can we avoid that? Because I see the strategy you use is to use a subunit. It's in a certain sense all subunits you're proposing. Can you give some comments on that? How we avoid these allergic reactions? So after infection, and naturally you vaccinate, and then when the massive infection is coming, it's worse than if the animals would not be vaccinated.
So that's a very difficult question. First of all, I think in that case, the big issue is that for the formalin inactivated RSV vaccine, that essentially did not induce almost at all neutralizing antibodies. So, and obviously they can provide the first line that reduces significantly the burden of viral replication if you have neutralizing antibodies up there. And that is believed to reduce the risk that antibodies and through complement deposition and effector function, once the infection is largely established in the respiratory tract, can trigger this kind of enhanced disease. The same, I have to say, and this has been just presented a couple of weeks ago in Madrid, has been seen recently in a trial in humans where they have tested an anti-stem monoclonal antibody for flu therapeutically. So giving the antibody when the infection is largely established, even in the lower respiratory tract. In this case, they did the study in hospitalized patients, and they actually found a trend for, not significant, but a trend for enhanced disease. But that does not mean that you cannot use the very same type of antibodies in a preventive fashion, because you can and this has to be proven. And I think from the animal studies, we have evidence of that, you can, by blocking the replication with highly potent neutralizing antibodies, you can block the spread of the virus from the upper to the lower, and by doing that, you can reduce the disease and the immune pathology related to that. And
so
there are also...
It's a concentration matter.
It is a concentration.
Antigen versus it's a dynamic interaction in antibody-antigen complex.
And
antigen concentration, antibody concentration, and then the complex.
But I think it's
a dynamic interaction.
It is, and it is key that the antibodies that are developed should be endowed with a potent neutralizing activity, so they have to be able to block the spread.
I know, but the ones they use for therapeutic treatment there, the last case you mentioned, was there a high affinity antibody or low affinity antibody?
Was a high affinity.
Because that is inducing immune complex formation, stable complex formation.
Yeah, I think these results have clearly indicated that there is a very low probability that passive vaccination, passive administration of antibodies, at least for flu, could work in a therapeutic fashion because of the risk related to that. Obviously, there is the possibility to do FC engineering to reduce the risk that this enhanced disease could develop. There are, as I said, mutations on the FC that can be introduced to avoid the binding to complement and others. But again, the truth is that we don't know enough.
Yeah. And then the last question. Okay, we were discussing yesterday with Lonneke and with a lot of people about this antibody complex constructed vaccines. If you have immune complexes, it appears that they are very good vaccines to induce immunity.
Yeah.
Now, we couldn't find out how it was really working, because if it is an antibody against a neutralizing epitope, then you will never activate these B cells which you have to activate to induce antibodies. So, can you give some comments on how this antibody complex antigen are good vaccines and how it works? Can you, give your idea about it?
Well, I think you are right that if you potentially have a masking effect of the antibody to induce antibodies against the target epitope on the antigen side. So that's quite possible. But I think it is true that immune complexes would allow to have a very efficient uptake of the antigen by phagocytic cells and a very good presentation and cross-presentation of T-cell epitopes, which I think is something that it has to be explored, and I'm working on that for flu by considering, in this case, some mutations in the FC that can tune a better phagocytosis by APCs for the presentation of T-cell epitopes. And in this regard, it is also quite important to consider that on B cells, there is a mechanism which is related to the presence of the inhibitory FC gamma receptor 2B that is working as a negative feedback on B cells to avoid the activation of the B cells that are specific for the same antigen. And so this is a well-known mechanism. But I think that all in all, and I was quite curious to see the data that were presented yesterday, that they believe that the pre-existing antibodies can have a detrimental role, necessarily a detrimental role in the immunogenicity of the vaccine is not that true.
That's what we experience in quite a lot of cases when maternal antibodies are present in chicken and pigs.
No, I mean...
Not always, Eric will, in some cases, not always, but...
I think that when you have a very high polyclonal response to the antigen, so you have the formation of big immune complexes, cross-linked, I think that's not the same as of having an immune complex formed by a monoclonal antibody against a specific epitope. And I think this is now also considered as a way of trying to boost the immune system, for instance, in chronic hepatitis B, because we know that there is a defect in antigen presentation and T-cell responses, and so we are hoping that by forming immune complexes with the S antigen, we can try to restore a good T-cell response. But it's very open field. So.
Thanks, David. It's a great summary of this area of work. So I think I asked you this question about 2 years ago, so I'll just ask it again and see whether things have moved on. And that's really relating to the frequency of the B-cell precursors for these broadly neutralizing antibodies. I mean, certainly the work with HIV suggests that they're very low frequency, and even though we may identify the antigens, is there any new developments in the prospect of being able to amplify those rare clones, essentially, so that they become high frequency and you get high titers of antibody? What immunization strategies? The first question is, now we have a lot more experience of these broadly neutralizing antibodies. Is that still true? I mean, and if it is still true, what's the likely strategies immunization strategies to increase the frequency?
That's very important. It has to do with the point that I was talking about here, the first one. So in trying to understand, first of all, what is the level of maturation of antibodies that we need to have high affinity? That's quite important. And on top of that, what is the potential frequency of precursors that have already a good starting affinity against the immunogen? So, as I said, for HIV, this is clearly an issue because the level of maturation that is required is very high. So sometimes you need 20-30% of the sequence which has to be mutated to have a affinity high enough and the breadth high enough to be defined as broad. That's very difficult. And in the case of HIV, they have found that there are some V gene families that are particularly frequent in all the BNAbs for HIV, and that's a good starting point. But the issue is that when they remove the somatic mutations from those antibodies, the antibody does not bind to the antigen anymore. So, and that means that probably the starting antibody in the germline status has a very, very, very low affinity for the antigen, or maybe it is triggered by something else. And the strategy they are trying to pursue for HIV is now to have engineered HIV envelope structures able to bind to the germline antibody precursor of this family of, in this case, VH1/2 antibodies. This is a very complex kind of approach in which they are thinking to develop a prime boost strategy in which they start with an antigen binding to the germline precursor and then trying to evolve and drive the evolution of the antibody response towards those mutations that have been found in the BNAbs from patients. So this is clearly very challenging. But this is not the case always the case for other pathogens. For flu, for instance, we have seen that in humans we have a very large frequency of precursors of antibodies, in particular in the VH169 family targeting the stem region. And there is a complication there, which is related to the fact that we have different alleles, in the population, and there are some people who have the good alleles to develop this response and some people who don't. That's on the side, and I think there is also a lack of information about the alleles in some populations, like in the African population. We don't know much about the V gene alleles, so I think there is a lot to learn there. But apart from that, what we have seen is that just 1 or 2 mutations are required for a fully germline antibody to be developed into a very high affinity neutralizing antibody. And I think that brings hope that with a relatively high frequency of precursor for anti-stalk flu antibodies and the requirement for just a few mutations to develop high affinity, this is a potentially an achievable goal as soon as the antigen will be properly engineered. And I think that the same applies also to RSV, in particular the Site3 antibodies. There was a paper recently published by Bernie Graham, have been found to be largely germline. So in that case, if you take out of all the somatic mutations, the antibodies are as good as when they are mutated. That means that there is a lot of the mutations that are introduced are useless. in terms of activity. Useless in quote, in the sense that they are important to stabilize the molecule, but they are not required for high affinity, but they have been shown to be required for increasing potentially the breadth of the antibodies. So I think it's case dependent, and I think that for respiratory viruses and for some more simple type of targets, not drifting too much, the frequency in the repertoire is high enough to develop antibodies with the high titers.
So a couple more things. So related to that, there's often discussion about when you're using structural vaccinology and you identify these epitopes that will drive broadly neutralizing responses, and having the idea of enhancing the presentation of those on the antigen, and one strategy would be to remove the specific, maybe immunodominant epitopes, so that the more broadly cross-reactive epitopes become more prominent in the antigen. Is there any success in that field?
Well, I think the headless kind of approaches for hemagglutinin are an example of this. It's not to the extreme. There has been also, which I didn't mention, and it belongs in this scheme to what it is highlighted on the right here, which is to have a grafting of epitopes into backbone proteins. But it has not been shown to be particularly successful, because when you do things like that, you end up developing a lot of antibodies that are not of the type that you would like to induce. In the case, instead of developing domains or pieces of protein like you're saying, this I think has a little bit more chances of success. So to the best of my knowledge, the most advanced approach is the one for HA on flu. There has been also attempts to do this in another way, which is to introduce a lot of glycans around the epitopes that you don't want to target. So this, in other words, to use this glycan shielding approach. But again, we don't have to forget that the glycans can be target as well of antibodies. But I think clearly that is much less advanced. And it's also important then trying to optimize the immunogenicity when these domains are isolated and stabilized, to display them in repetitive units like on VLPs, so to potentially induce the level of immunogenicity. But we are still, I think, in the very early days of this kind of engineering.
So one final question, and it may be slightly outside of your field, but in that it's and the other major challenge is driving duration of immunity, increasing the duration of those antibody responses. I mean, any advances in the strategies to try and do that with antigen design?
Not that I'm aware of. I think that has really to do with finding out the best adjuvants to be used. And those that can induce the most persisting immune response. But I think that there is also the big problem there that we know well that some adjuvants work well in some animal species and not in others. So how can we really translate well what we learn from animal studies into human studies? Is not easy. It's true that there are now new adjuvants that seems to induce a more type of sustained and a higher level of response, but I think that in a nutshell, this looks to me yet very empirical.
So the question, I mean, adjuvant's fine. I was thinking about antigen design. So the striking example of the conjugate vaccines for meningitis, etc. That the conjugation process clearly had a big impact on the duration of immunity.
On the T cell side, that somehow could also be linked to the ability or to try to boost with this type of subunit vaccines a better T cell response to the very same antigen, and One possibility, as I said before, is to try to make immune complexes, which could be a way, but...
Also live subunit, if you could produce a live subunit which continuously produces the antigen, like naked plasmids and such things, your stalk region, then you would have a continuous supply and perhaps a continuous affinity maturation of your B cells to select for the good antibodies.
Yeah, no, it has been explored, the possibility of grafting like the stabilized RSV antigen into viral vectors that are producing the antigen actively or through the DNA, this DNA approach and so on. But it has not yet been clearly proven whether they end up to induce a more sustained response. So
I had a question about the nanobodies. Oh, sorry.
The nano?
It's my, if there are other questions, other people can answer.
I have a question, only one. So Davide, first
You
of
only
all,
arrived today, eh?
yeah. Davide, first of all, congratulations for giving us such a beautiful overview of designing vaccines in the direction of neutralizing antibodies. But I'm working with PRRS. I see that you work a lot with human viruses. PRRS is an animal virus, so I don't think you know a lot about those viruses. But this one is really a one that is escaping from all branches of the immunity. And also a lot of the viruses that you give are RNA viruses that are very unstable. So my question to you is, if you know, because I see that a lot of scientists want to create kind of small subunits and things like that. Of course, you will induce neutralizing antibodies, but what is the long-term effect of of those subunits? Because I think your virus will really, in evolution, will get away from it. So, what do you think about that? And isn't there? I think there should be more a kind of a strategy that people should know, that you should activate a lot of branches of the immunity, not having problems with that force of evolution. And I see a lot of people working with subunits, and I really would like to stimulate them to think more in directions of vectors, so to really activate a huge variety of responses, so that you don't put a lot of pressure on your neutralizing antibody, on the ligands that are inducing neutralizing antibodies, because in long term, I think it will be difficult to keep that thing as a real antigen. What do you think about that? And
you are referring to the risk that due to the drifting of the pathogen, you can have an escape issue. So, well, I think one of the reasons for flu to target the stem is clearly to try to target a region which is relatively constrained in drifting for the functionality of the pathogen. So there is quite a lot of work which has been done in the last few years in trying to define whether the conservation of the epitopes targeted by the stem antibodies for flu is due to the fact that there is no pressure for evolution on that region because of low level of antibodies, or whether there is an invariance due to the fitness cost for the pathogen. It seems to be more the second for that specific region. So, and that suggests that if we can successfully try to focus the response on that region, the risk for flu of escape is honestly pretty low. I don't think that is the case for RSV. Clearly, which is a virus that is known to drift much less than flu in terms of evolution over the years. But in that case, we know that there are some regions that are that are targeted and that induce highly potent antibodies that are prone to mutate easily. So I think you are right. That is potentially an issue, and it has to be. It is also obviously a big issue for the development of an HIV vaccine. It is true that we are dealing with pathogens where, like HIV and RSV, for which we don't have a vaccine. I think at least we would like to start from having something that works to some extent, and then we can build up on that. In the case of influenza, instead, clearly the goal of the vaccine development is to develop what they call a universal type of flu vaccine, with a lot of misunderstanding on what universal vaccine means, because universal to me does not mean to induce antibodies that can cover for 3-4 years and then the virus is drifting. It's clearly more than that. And I also agree that we need to try to develop strategies to induce a much broader type of immunity, where we will also include to have very broad cross-reactive T-cell responses that can be very important, and they have been shown to be. I'm working a lot on antibodies, but I'm very well aware of the importance of T-cell responses in controlling the disease severity. So I think that is clearly very important.
And one question to follow on that line, because in fact, even for RSV, the correlates of protection are not so clearly established, and neutralizing antibody as they are measured in vitro, they may not really mimic what is really happening in vivo. And for instance, a nasal IgA in human or nasal IgG2 in bovine may be very, very strong correlate of protection. So my question is, are you also working on that aspect? Do you have you possibilities to isolate broadly neutralizing antibodies, but IgA1, or do you work on that or not at all?
We are working on IgA, not for respiratory pathogens. We are working on IgA for enteric pathogens. We have a collaboration with the Gates Foundation for the development of secretory IgA antibodies. But coming to that point, I agree that there are some good correlates of protection with like nasal swab IgA and RSV. And I think more recently for flu, it has been also shown in some studies that was just published a couple of months ago that anti-neuraminidase antibodies for flu represent another very good correlate of protection in human challenge studies. However, I think that we have also to be careful to be sure that we are looking to the full picture, because this can be direct or indirect correlates. In other words, for instance, for the neuraminidase antibodies, it has also been shown recently that influenza vaccine does not induce really good titers of anti-neuraminidase antibodies, whereas the infection does. So I think we also need to understand whether what those correlates of protection are related to something else, like in this specific case, also the fact that the infection could induce neuraminidase antibodies, but the infection induces also good T-cell responses. And to try to answer to your question, I think we are interested to have a better understanding of correlates of protection. We are developing some of these monoclonals into the clinic, and I would like to take advantage from having clinical samples from our studies to have a better understanding on which are the humoral and cellular correlates of protection from these studies. Obviously, this is not easy, and it requires a lot of sampling and the availability of a lot of tools. But I agree that it is of utmost importance to have a better understanding, and that neutralization is just a piece of the story. I completely agree.
David, can I just ask a question? You've explained a lot of complexity, and obviously this is sort of making advances in human health. Is it feasible to imagine that this kind of work can be done in the animal health sector, where vaccine price is actually pretty critical? What is the cost of actually taking this technology and putting it into
that's very interesting. I am completely aware of the fact that the cost issue, and this was very well outlined yesterday, is very important for the development of animal vaccines. But I was wondering, in the case of the RSV vaccines that we develop, and we work with Peter Kwong and Geraldine, in that case, honestly, you have seen the results. So with 50 microgram per ml, we got a pretty robust response. It's really not a big issue to make a stable cell line producing 1 to 2 grams per liter of the very same antigen. So we have the technology to do that. So the cost will be a few cents per vaccine. So as soon as obviously the level of purity of because most of the cost comes from the downstream process, so the need for high quality kind of GMP type of material. But if the requirements are, which I guess is the case, are not that high for animal vaccines, I think that there are the technologies now out there to reach high yields with this kind of approaches in the production process to end up with the cost of, I don't know if we can call it cost of good for vaccine, but which is definitely reasonable. And I'm still wondering why there is not much interest from the field to go this way. I mean, I can imagine that the cost of attenuated strains is relatively low, but I'm wondering if it is, if we are not more or less at the same level. Obviously, the complexity which is behind the development of these things can be difficult to be applied for all targets, but I think that we should consider that seriously as a valuable approach. Because as far as I know, this is not really typically the standard in animal vaccines.
Well, I mean, I have to say we've been working for the last 10 years on using structural vaccinology to improve FMDV vaccines, and we've got stable and using the same space-filling mutations, cysteine bridges, etc. I mean, this is all published. So we've got very stable FMDV empty capsids. The level of production of those now is compatible with commercial production. We'll probably end up with a product cheaper than the current vaccine. So yeah, it's there's a large body.
The answer is that indeed we can do that at a reason.
It's perfectly feasible. And it's insect cell production and that sort of thing. It's easy, yeah.
But is
easy.
the industry? Sorry, this is a question to you. Is the industry ready to in developing?
Definitely. Oh yeah, they're fully on board. I mean, you have something.
Because I was surprised that when we were looking around for some companies with potential interest for this bovine RSV vaccine, there was really no.
Well, is that? I mean,
there'll be
better. Maybe Andy's better to answer this to me, but I think a problem with the RSV vaccine, you've got to displace another product from the market. That makes it more challenging.
I was just, sorry,
No,
reflecting on respiratory vaccines for calves. And in 1972, I was injecting calves with a product called Pneumovac Plus, which contained it was a multivalent viral vaccine to prevent respiratory disease in calves. And here we are, what is it, 45 or 46 years later, we still don't have an effective RSV vaccine. And so I would wonder what the appetite is actually in the animal health industry to do that really.
My guess would be it would be to a company that doesn't have a product rather than one that does. Just a guess.
But then there are patent issue and all those type of things also. That's complicated.
Yeah, but you've got.
There is a patent on
freedom
this,
to operate on the IP.
Yeah.
Yeah. So that's a good thing. The IP's owned. Yeah. So you'll find me. Speed dating.
Yes, let's talk about it. I mean, but I forgot to say, but the results from the bovine study were very, very well taken because of the relevance of the model, as a suggestion that the very same type of approach could work in humans for RSV, and that's why. I think, as I said, two pharmas are moving forward, and maybe we will get a successful RSV vaccine in the next decade. So.
Okay, I think we can go to coffee break and further discuss about that.